Hello, everyone. Don't be so serious. Keep drinking. I want to thank Ali for the very generous introduction, and it's my great pleasure and honor to deliver this speech here. When I asked Ali what kind of talk we're expecting, and he said it's an after-dinner talk. So using his own words, keep it light and entertaining. So that is absolutely a tall order, so let's see. Okay. So as Ali said, I've been a professor in the Department of Biology for over 20 years. And my laboratory studies how genetic informations help to wire the cells in the brain. Okay, and then that is my one hat. But tonight I will be mostly wearing the other hat, which is that I'm the director of the Wuxi Institute. And here, this is the physical location of the Institute. We have over 500 faculty affiliates, 7 schools, and 50 departments. I'll say something at the very end about why so many schools and so many departments. Okay, and this is my attempt to be entertaining. So brain is the last frontier of biology for 2 reasons. The first reason is that just about everywhere else in your body, when you think about biology, you can pretty much distill it down to biochemistry. 2 things interacting with each other, 2 proteins, lipids interacting with each other. That's biochemistry. We can make sense of what happens in the rest of our parts of our body with biochemistry. But in the brain, we're dealing with questions such as how is consciousness generated? How do we learn from experience? Where do are our memories stored? These are questions, key questions in neuroscience that we just simply cannot be used biochemistry to explain. Okay, so that's the unique aspect of neuroscience, is that we're still very far from really understanding all of these very key questions. That's the first reason. And the second reason is that the brain is just incredibly complicated. We have estimated 100 million neuron cells in our brain, and they're connected by on the order of 100 billion synapses. So these are incredibly astounding numbers that we need to dissect. That has been sort of the obstacle for understanding brain science and neuroscience. So I thought, how can I be entertaining? And the goal tonight is that maybe in a few months, you could still remember a thing or 2 that I said. That would be a success for me. So here I'll start by asking a question, discussing about is IQ inherited or not? And then I will then pose the question whether you think rodents can do geometry. You know, I picked these topics so that based on their entertaining values for engineers. And then at the end, is a bionic man like Tony Stark? Well, the Marvel movies has really made my kids believe that Tony Stark is for real. So, but is that still a pipe dream, or is that something that is about we're on the way to? So joking aside, these are the topics that I will cover: neural mechanisms of spatial navigation, digital twin brain, and artificial retina. And all of these research are ongoing researches in the Neuroscience Institute at Stanford. So I'm not talking about my own research. I'm talking about colleagues who have been funded by Wuxi Neuroscience and then in our institute. Okay. So here I want to take a moment to ask you, IQ, right, is a measure of intelligence. It's debatable what exactly is IQ measuring, but nevertheless, it's a quantitative way very widely used way to measure intelligence. Let's just, for today's discussion's sake, you know, if you look at the distribution of IQ within the human population, it displays a normal distribution with a mean right around 100, with a standard deviation of about 15. This is data from 10,000 individuals measured. So I want to take a poll in the audience. Who do you think IQ has more to do with genetics? Your mom and dad. And who do you think IQ has more to do with the environment? Which school you went to? What was the first age that you were exposed to two different languages? Things like that. So for those of you who thinks that the genetic information plays a more important role, please raise your hand. Okay, I see a lot of hands. How about the opposite camp? There's a fair camp. Great. So here is a lively debate about which one plays a more important role. How do we, this is clearly a function of the brain, right? But how do we even approach this, right? So although we don't know the neural basis of intelligence, scientists has approached this using a very interesting genetic trick. So here's the following. So if you were to take that 10,000 individuals and randomly pair them up, form 5,000 pairs, and their IQ values are not correlated with each other at all, right? Your IQ has nothing to do with the person sitting next to you's IQ, okay? Makes sense. If you now look at 5,000 pairs of monozygotic twins, maternal twins, then their IQ value is highly correlated with a coefficient about 0.86. So you would look at this and say, oh, it's genetic factor. But that's not a proof for genetic playing a very essential role, because these monozygotic twins very often, if not always, are raised in the same environment. So their environmental conditions are also very similar. How do we control for that? We look at dizygotic twins. These are paternal twins. They share the genetic material with each other, but not identical genetic material, okay? So the dizygotic twins correlation is about 0.6. And you can basically do a calculation based on this equation. You can calculate the differences between the monozygotic twins and the dizygotic twins. Of course, the use of the dizygotic twins is to eliminate the non-genetic information or to reduce the genetic information. And here, after this calculation, the answer is 0.52. So about half of IQ scores, it comes from genetics. The other half come from non-genetic. So everyone is right, or everyone is wrong. Right. Compared to other features of our body, height, our physical features are almost entirely genetic, and major psychiatric and neurological disorders have the heritability between 0.3 for major depression and then 0.7 for these two other conditions. All right, so I just wanted to give this example to how the way to think about this is that does genetic contribute to our central nerve, to our brain? Of course it does, right? You can. Our closest ancestor or evolutionary neighbor is Great Alps, is chimpanzee. Chimpanzee will never learn a human language, right? Although chimpanzee and human are very similar, if you look at the main part of the genome, they're very similar, but they will never be able to learn human language, while all humans can learn our language. So in some ways, you can think about this as hardware versus software, right? The genetic information is the blueprint that builds the hardware. And then your training, your experience is the software that can write onto your hardware so that you can become the computer you are. All right, so that's just one example of, okay, so we can analyze, we can study neuroscience from this behavior level, and we can use genetic tricks to make some rough dissections of whether it's nature versus nurture that affects our brain. But to really understand how the brain works, you really have to know how the hardware is built and how the software is written, so that how the brains can encode information. So here I'm going to use a beautiful work from Lisa Giacomo, who is a professor in the Department of Neurology, also in our Stanford Neuroscience Institute. And her lab is interested in understanding neural components that builds a cognitive map of space. So what do I mean by that? A common experience that every one of us have had is that we walk out of a parking lot, so now you have to find your car, right? And finding your car, meaning that, if you think about this, means that you have to have a memory of where you parked. And if at the moment you walked out to either Walmart or Disneyland, you have to have a spatial map in your brain that represents the space. You have to somehow extract information from your memory of where my car is, and then according to the map that you instantly built, you have to plan a path towards that car. And every moment during your movement, you have to update this map so then you can ultimately get to the car, right? So that's a very complicated thing to do for a brain to do, yet every one of us can do it. Probably not to the same extent. I often got lost about this. Okay, so humans use landmarks and they update their positions in this space, and then they go, they plan their path of traveling. So how does this work? All right, so it turned out that one part of the brain that appeared to be very important for this task is deep in your brain. There are these blue regions called medial entorhinal cortex. It's part of the cortex, okay? And here is the experiment. This is probably the most complicated thing that I'm going to say tonight. So here is the experiment. In this little blue part of the brain, there are many neurons. And what Lisa's lab has done is they stick an electrode or multiple electrodes in this region. And then they wake up the mouse. Okay, this is a little surgery. They implanted that electrode into this part of the brain. They're recording from, let's say, individual neurons in this area. And then they make the mouse explore this 1 meter by 1 meter arena. And then they are recording from this neuron. They're reporting when and where this neuron gets excited. So when you see a red spot here, that means this neuron is screaming, it's very excited, it's activated, it's firing. And when you see blue spots, that means this neuron is quiet. Okay, so they let the mouse run around here for a while, and then here are the spots that this particular single neuron is firing like crazy. And remarkably, if you notice that this neuron doesn't fire anywhere on this map, it only fires in these spots. And these spots are not random spots, but they form a grid. So this grid is not a rectangular grid, but instead they're equilateral triangles. Hence, although rat cannot do geometry, their brain can do geometry. So the activation of these neurons are based on their visual cues, right? When the mouse is sitting in this part, in this arena, its visual cues tells it that, okay, I'm in this arena, but without them knowing it, their cells are firing at a grid, like coordinates. Every time the mouse is here, this particular cell is going to fire. And it's going to fire also at this distance away from it. Okay, it's pretty remarkable. These are called grid cells. Okay. Next time, and there are two features of the grid cells that you should know. One is that there are different grid cells that are firing at different positions, but their organization is very similar. So that's very robust, right? I'm standing here. One set of grid cells is going to fire. Now I'm standing here, another set of grid cells is going to fire. Okay, that encodes the physical locations within the room. Another feature is that when you go to a slightly different part of the brain, the grid cells changes their density. So that means, their density, they're still grid cells, they still are equilateral triangles, but their distance, their density is slightly different. So this provides even more rich information for you to encode the position of that individual animal. And then they found that there are additional cells. For example, this cell, this is exactly the same experiment, but they're recording, they're listening in from a different neuron. And they found that this is a border cell. So it only fires when the animal reaches a boundary of the arena. So this is thought to, when you see a wall here, you know where you shouldn't be going further than that. That fires, another neuron fires like that. There are head direction cells. So only when the animal is here, only when it's facing this direction, it will fire. And then there are speed cells, where its firing probability is proportional to the running speed of the animal. Okay, so now you have all of these informations. It basically, all of these cells are in the blue area. They are ultimately drive cells in the hippocampus, which is in this little yellow area to form this place cell. This place cell is only fires when animal is in a specific location, only that location in the arena. So, there's still many questions that are left that we still don't understand, but we've found that these particular areas of the brain, Lisa's lab, along with her colleagues, that basically are showing a representation of a room that you're in, right? So if you think about these in the animal world, right, we need to have, the animal its ability to understand its environment, knowing its location, is very important for its behavior, especially for important behaviors such as getting a pot of food, or finding mates, or escaping a predator, right? So it seems that it would be more advantageous to be able to change the map upon the location of reward, safety, or food. So indeed, the Giacomo lab did this very cool experiment where they basically had the mice explore the same room, but in a second room, they put in this food reward. And they found that the environment 1 is without food and environment 2 is with food. They found that when the animals are getting reward from a particular location, they tend to allocate more grid cells to that location. And they think that this explains, the way to think about it is if you have a rat that is exploring an environment such as the San Francisco Zoo, and they have a representation of where they are, build a spatial map in their brain. But one day, when they found a big piece of pizza there, so they would want to remap their cortical representation so that to have a finer map in that location so that they can go back. So not only do we have grid cells that tells you serve as a universal metric system, but this is a flexible metric system. It can be trained by experience, by a reward. Okay. All right, so it's worth mentioning that actually this entorhinal cortex and the existence of these universal metric system appear to be evolutionarily conserved. It is found in fish, flies, bats, rodents, monkeys, and even human. So it's how this very primitive part of the brain is representing the external world, the spatial aspect of the external world. And here is Lisa's lab. Lisa is right here, and she trains her graduate student postdocs. They are electrophysiologists, but they got a lot of help from people like Surya Ganguly, Scott Linderman. These are modelers. They're basically AI engineers. They crunch data. They also build models. And then it's becoming more and more common that we need collaborations between engineers and neuroscientists to really make progress. And I'll emphasize that point again in the next example. So here, what I just did is to focus on this little part of the brain that seems to be encoding the spatial localization, but the brain is really connected, right? All our brain cells is one way or another connected to each other. And here, so one of the biggest hurdles in our understanding of how the global brain functions is how is the astounding number of connections and the scale while we're studying like a single cell recording, there are millions of cells around you. You don't know how, when you're recording, listening in from this one cell, what happens to all the other cells. So here, our very distinguished colleague and my friend, Dr. Jin Lee here, who is here at my table. And she's really the pioneer in understanding, bridging these scales between a cell-to-cell communication, a very micro scale, to a much larger mesoscale brain functions. And then she's an engineer. She uses functional fMRIs, and she came up with a very interesting way to stimulate neurons using optogenetics. And then she applied that optogenetic stimulation to understand how the greater brain functions. And she has understood the brain, so she's built mathematical models taking into account of the biophysical property of each connections, and ultimately built into a model that can predict a lot of the behaviors in microcircuits. So here's the actual data recordings. When these neurons fire, you'll show a cluster of spikes, and then her models can actually can perform quite well compared to the actual data. So the take-home message here is that in addition to experimental recordings that we can measure detailed communications between a set of neurons within a circuit, there are now mathematical models that can be used to study a much greater scale, including many more neurons. And that is really, we've started to get tractions in this using modeling to understand how brain functions. So I want to change gears here to talk about neurodegeneration, and this is a major challenge for our society. For Alzheimer's disease alone, this is the most frequent or frequently occurring dementia. 1 in 20 people is affected for over 65-year-old population. But by the time the population reaches 85, half of the individuals will be affected by Alzheimer's disease one way or another. 2 out of the 3 centenarians have dementia. So these are astounding numbers that just tells us that our brain will deteriorate in an age-dependent manner. And then that is already a well-established fact. Sorry for the bad news. But why is this the case? You might ask that question. There's several, of course we don't know all the answer to that, but there are several several factors that I think for sure will contribute to that. One is that the neurons do not regenerate. Unlike our skin cells and like our muscle cells, you go to the gym and you exercise your muscle, you break the muscle cells, they die, but their stem cells they stimulate the stem cells to produce more muscle. That's how you grow your muscle cells. You get bigger, right? So, but unfortunately, neurons don't do that. We have the neurons that we're born with. Okay, when they die, they die. There is no generation of new neurons, very little regeneration of new neurons in our brain. Okay, so that is intrinsically a problem, it's a huge problem that we don't regenerate neurons. A second big problem is neurons are demanding cells. They are large, so here are the drawn to scale muscle cells or epithelial cells, so some of the small blood cells. And here you can see the neuronal cell bodies has these antenna-like dendrite that receive signals, and then they have these extraordinarily long axons that can be as long as 1 meter long. So there are cells that live in the back of our brain that controls movement to send all their single axon down our spine. And this thing can be as long as 1 meter, and that's huge compared to the microscales of molecules. So because of the long processes, there are molecular motor proteins that are trafficking on the microtubules. These are the intracellular highway systems, and this is of course like hugely magnified. You can see that they're dragging materials down this highway, and of course that consumes energy, and anytime the highway breaks, anytime the motor breaks, over many decades that these cells are with us, then we're getting into troubles of neurodegeneration, right? That's the two factors to consider, but of course, you can ask why. I'm sorry to deliver this even worse message to you tonight. Our brain is not designed to last for this long, okay? We are here, we're the way we are because of evolution. So think about this, right? Evolution has selected us to be successful as a species up to the point our kids grow up to their teenage, they become independent. They don't want to listen to you anymore. They are on the phone all the time. Tell me about it. But then our value in a greater sense of evolution and biology has diminished. So effectively, we have a car that is designed to go 200,000 miles, and now we're asking it to go 800,000 miles. So that is what we're dealing with here. Okay, so it's very difficult. Our neurons are probably going to degenerate over time. Okay, so how do we deal with it? Of course, extending lifespan, extending brain health span is a huge challenge, and it's extremely. People are so interested about this, right? Once you realize that, whatever we can do to extend that healthy cognitive function is priceless, right? So we are very fortunate to have received a very generous donation from the Knight family, the Penny and Phil Knight Foundation, and then within the Wu-Tsai Institute, we have established, just starting last year, this Knight initiative, focusing on advanced science to extend healthy brain and to slow down brain aging. Okay, we're taking all kinds of very interesting ways. I'm not going to talk a ton about that, but you're welcome to ask me about that. And I'm just going to use one example this is the rest of the talk, to show you one way to think about how we can use engineering to build bionic people to combat degeneration. So here, of course, I'm talking about artificial retina. So the macular degeneration and retinitis pigmentosa are conditions that affect many people. So 170 million people worldwide these are conditions that leads to blindness. Okay, in this disease, what happens is that the photoreceptor cells die, and then the individuals cannot see anymore. And then so our goal is to restore vision instead of replacing vision with the stick. And so this is how the retina works, is that there are photoreceptors that has rhodopsin, has opsins that absorb lights and then convert light into electric signals. And then these signals are analyzed or processed by a group of cells. Here, there are amacrine cells, bipolar cells. There are a bunch of neurons in your retina. And ultimately, these signals are sent to the retinal ganglion cells. These ganglion cells then are the only output out of the retina. So they send the information into your brain. Okay, so the retinal ganglion cells is the output cell. Okay, and in these neurodegenerative conditions, such as the macular degeneration that I talked about, these photoreceptors died. Okay, the very upstream, the sensors died. Okay, of course you can't see without the sensor. Right? So the question is, can we bypass the sensor? We have good camera chips. Can we bypass the sensor and sense the information directly to the retinal ganglion cells and therefore restore vision? That's the challenge. So this actually has been an idea that has been tried in the past. So for example, this is the first sort of generation of chips with 64 electrodes. So these electrodes are implanted into the retina, and then it was driven by a camera. And this is the first generations. It's called the Argus One retina implant. It was approved by 2013 and then discontinued in 2019. Okay, and the reason it got disapproved or discontinued is because it doesn't work very well. So it gives the patients a sense of flash of light, but it doesn't really give you a good, well-resolved picture. And why is that? So the reason that it failed is because our eyes, our retina is not a camera. Okay, so cameras are pixel-to-pixel representation of the visual world, two-dimensional world, right? But our retina is not. Our retina transform these two-dimensional information, extract that information, and turn it into a bunch of spikes in the retinal ganglion cells. Those spikes are getting sent into the brain. So while there is the two-dimensional distribution of the retinal ganglion cells, it is not nearly a pixel-to-pixel translation. So the challenge is to understand this transformation from the two-dimensional visual stimulus to this code. Okay, that is the challenge. So how do we do it? This is the work of a team that's led by E.J. Chinchinitsky. He's a professor in electric engineering at Stanford. So the approach that he took was to design a set of smart electrodes. Okay, so the way that he does these experiments is that he plants these high-density electrode in the retina, but unlike the first generation, what he does is that he first listens in on spontaneous activity to see which of the electrode is next to what kind of cell. Because each of these ganglion cells are different from each other, so they fire with signatures. So he put the electrodes in, and there are many electrodes, high-density electrodes. So he first listens in. Based on the intrinsic properties of these cells, he knows, okay, electrode 1002 is right next to this type of cell. So he took the biology that we have understood from studying the retina from the retina to really to guess what kind of cell that is. That's the first step. And the second step is that he stimulated that electrode to see what is the optimal way, what's the way to stimulate that cell type. Okay. And then the third step is that he uses pattern images to see to come up with the algorithm that can transform that image into the neural code. So basically replay that image through the understanding of that transformation to activate the retinal ganglion cells, not in a pixel-to-pixel transformation, but the transformation is what really makes the difference, right? Okay, so this is, of course, easy on the paper, but it's very hard to do. So this includes the fabrication and designs of these high-density electrodes, and also build this power supply or communication apparatus that talks to the electrodes. So this is wireless, of course, wireless drivers. So you're basically carrying a little computer on your glasses, and developing the algorithms that are really transforming the two-dimensional world into the neural code that can be played back to these retinal ganglion cells. Here is his famous experiment where they use his monkey ex vivo retina to try to actually record from these ganglion cells to understand which of the electrode is next to what kind of cell. And this also involves, so now they're really at the stage of putting it in human, so they need to involve neurosurgeons where they can fabricate these long electrodes that penetrates the optic nerves, and ultimately come up with a stable transplant strategy to put it in human. So this cannot be done with EJ's lab itself, and as you can see that it actually involves people from electrophysiologists that he trained, but also engineers, computer science, data science people, and people specialized in electric interface, but also from physics, from ophthalmology. So it really takes a village to get this done, because this is way beyond what a single lab's expertise can cover. So here we're very proud that the Wuhan Neuroscience Institute funded this project and really brought these people together. So I take pride in, I didn't do it, it's my predecessor, Bill Newsom, who really started this project. So thank Bill for that. This is what I covered today, and I hope you'll remember like one thing or two in a matter of a few months. But I want to end by thanking Matthew Scott. Where's Matt? Okay, Matt is there. Matt is a professor in developmental biology at Stanford. Matt is the funding director for the BioX Institute at Stanford. It was Matt's vision about interdisciplinary collaborations and Jim Clark's generous donation that really has spearheaded this entire mentality of bringing engineers and biologists and medical physicians together. And then I think that has really, we have been drinking that Kool-Aid and we have been copying that for the Stanford Neuroscience, and that model has been replayed in many, has been demonstrated successful in many leading institutes around the world. Thanks, Matthew. I don't think there is a question session, is there? I mean, I'd be happy to answer questions. I'd be happy to stay later to answer questions. Ali, should I answer questions or no questions? Yeah, I have my clicker. That's right, I have my clicker. I see a hand in the back.
Can you crank up the amplitude to overcome some of these degeneration aspects?
Yes, I believe. Again, I'm speculating here. I'm not one of the team members. This is a slightly outside of my expertise area. But I think you can. And then the fact that you actually want to target these individuals before their photoreceptors go completely gone. So you want to use the remaining photoreceptors to help you to map out what kind of ganglion cells your electrodes are placed at. And yes, so one of the power for if you have the electrodes is you can crank up the amplitude to amplify the signals.
So is the transformation from the signals to what is the machine learning model, and is it like a difference?
Yeah, I'm sure there are many components of machine learning embedded in the discovery, and I can't tell you the details. I kind of wanted EJ to come. Unfortunately, he couldn't come today, but he would be the person to ask that question. Yes. Is there? Okay. Thank you so much.